Leslie is a pediatric neurologist from the University of New Mexico in Albuquerque, who will talk about something that we haven't heard about. And it's some enzymes that are very important in inflammation in the spinal cord and brain. Thanks so much.
Thanks. Thanks, Doug. I wanted to mention a few things. Doug already mentioned I'm a child neurologist, so I really come to this as a clinician because we see 3 to 5 children per year with transverse myelitis in New Mexico. Cody Unser happens to be one of them, who came a couple of years ago and who has, as you know, been very involved in getting these centers together. I think we have several aspects clinically that we have to take care of. One is the emergency room management of patients, especially our very youngest children, some as young as 5 months of age at onset, and also the treatment that we can provide after we make the diagnosis. But I think we have a responsibility in the universities as well to think about what other kind of research we can offer. So yes, we want to be part of the transverse myelitis working group, but we want to offer something too. And so I had a talk with my chairman when the Cody Unser First Step Foundation approached us, and he has a laboratory which has been involved in looking at matrix metalloproteinases. And I'm going to tell you what those are because I've just been learning too. So we're going to learn this together. He had recently published an article with Raoul Mandler looking at DeVix and multiple sclerosis, and he said, why don't we look at transverse myelitis? So we're thinking of 100 or 200 samples of spinal fluid as opposed to 3 per year. So that would be exceedingly helpful to us. What is the matrix? There are 4 major components to the extracellular matrix. This is obviously means outside of the cell. These components are collagens, proteoglycans. These are all words that I don't expect you to really use around the house. Non-collagenous glycoproteins, and then elastin fibers in the skin and aorta. And I'm sorry, but in the handout, the references did not come out. They were on the slides, but sometimes they are deleted when you try to make things small. Extracellular, yeah, they're here now, but they're not on your, I don't think they're in the handouts in the book. The extracellular matrix performs a function of filling space between cells. It builds a scaffold for cells, providing structural integrity for some tissues, and this includes, in a sense, the brain tissue and spinal cord tissue. It provides support for cells. It's actually a very active substance, and what it does is it sustains cell adhesion, in other words, the stickiness of cells. That's very important in communication from one cell to another. And it also helps with proliferation or the division, making new cells, and also the differentiation of cells. So what cells decide that they're going to be. That's become very popular recently because of stem cell research, because stem cells are basically cells that don't know what they want to be yet. So it's very important that you can maybe make a difference in what these cells will do. Reservoir for growth factors, and people have mentioned today brain-derived neurotrophic factors and some of the other growth factors that might be important in development of new cells. So that's the matrix. What is a matrix metalloproteinase? Matrix metalloproteinases, or MMPs, and you can think of them as the military police. Zinc-dependent endopeptidases. The zinc is where the metallo comes from, and endopeptidases are enzymes which digest proteins. They digest the proteins in the extracellular matrix. So again, remember, that's your scaffolding. That's what's supporting the cells in your spinal cord. They're secreted as what are called zymogens, and that's how we assay them. We can look for zymogens in the spinal fluid. And they require processing by cleavage, or breaking up part of them in order for them to become activated. They're involved in physiological processes, which include wound healing. It's very important to us in spinal cord injuries. They're also in some other interesting literature. When I did a search for matrix metalloproteinases, I found a lot of literature in the OBGYN information, and also in angiogenesis, which is an interesting subject to me because of my work previously in cavernous malformations mentioned by Dr. Mandler. Because when you have an injury, these new blood vessels, if they form, can get in there. But they're also involved in pathological processes, and these include tumor cell invasion. So a lot of the literature in matrix metalloproteinases is in tumors, including brain tumors. And then in inflammatory diseases like multiple sclerosis. These proteins are a gene family with 20 members that comprise 4 major groups which differ in protein structure and in substrate specificity. They're synthesized, they're made by glial cells. And glial cells are sort of our support cells in the brain and spinal cord, but they're also very active cells. They do a lot of things. We used to think they just kind of stood around, but they don't. They affect the integrity of the extracellular matrix. So your support, your scaffolding is going to be affected by these glial cells when they make the matrix metalloproteinase. They may affect leukocyte entry into the central nervous system. And this illustrates that where you have an activated T lymphocyte, it then produces a matrix metalloproteinase. And that then allows the monocytes, or some of the blood cells, to actually go across the extracellular matrix by sort of dissolving it and through the blood-brain barrier. So now this monocyte is in an area where it doesn't normally go. And with that, this illustrates myelin, which is on the axon inside the brain, which is kind of appearing and disappearing. Major matrix metalloproteinases in the brain include 2 major ones that I'm going to speak about today. Gelatinase A, or MMP-2. I don't know why they have A's and letters and numbers. And then gelatinase B, which is MMP-9. So I've illustrated those in orange. Here's what they actually look like. The gelatinases A and B are this particular one, and they look very similar in structure to some of the other collagenases, those that break up collagen, matrolysine, which is involved more in the OBGYN tumors and development. So MMP-2 and MMP-9 are the ones we're really talking about. These enzymes attack the basal lamina surrounding blood vessels, and it alters the permeability. So what we're really talking about in a lot of these immunologic conditions and autoimmune conditions is that things get into an area of the brain which is supposed to be preserved. It's never supposed to see some of these things. But when the blood-brain barrier breaks down and when the extracellular matrix is broken down, these things can then enter into this sovereign territory where they're not really allowed. Normally, though, we do find MMP-2 in cerebrospinal fluid, and we find MMP-9 only when there's an inflammatory response, and so that normally you don't pick up. MMP-9 comes from T lymphocytes, and you've heard a lot about T lymphocytes from Dr. Irani, a lot of humoral by Dr. Lucanetti. So our immune system uses MMP-9 to attack the capillary basal lamina and allows crossing into the blood-brain barrier. There's an experimental model mentioned several times previously called EAE, and in this model, MMP-9 is increased in the rodent brain. So we kind of think that there's something similar in human disorders like MS. Inhibitors of MMPs block the manifestations of the disease EAE in animals. So that becomes very important for the role that we might play in introduction of inhibitors to this substance. MMP-9 is elevated in CSF in multiple sclerosis. So I told you it's elevated in EAE. It's also elevated in MS. And it's increased before an acute attack. So if you could assay this, can you give an inhibitor and prevent this process of breakdown? These MMPs, 2 and 9, are also called gelatinases. They're increased in the CSF of MS patients with gadolinium-enhancing lesions on MRI. So this indicates more activity within those lesions. So a treatment trial has been given in which high-dose steroids were given after measurement of the MMP, and they returned the MMP levels to normal in MS. MMPs and adhesion molecules. Now these adhesion molecules, I talked a little bit about the stickiness of cells and how that pertains to how they communicate with each other. These MMPs mediate the destruction of myelin components, and so we want to preserve our myelin, especially in transverse myelitis. Pro-inflammatory, so cells which cause inflammation, produce cytokines, one called TNF-alpha, and that's involved in the shedding of the cell surface molecules. A chemical called merimostat, which is a broad-spectrum MMP inhibitor, inhibited TNF-alpha mediated release of the soluble vascular cell adhesion molecule 1 in human cerebral epithelial cells. MMPs are actively involved in shedding of adhesion molecules at the blood-brain barrier. So really what we're really trying to figure out is, what are these things, matrix metalloproteinases, and are they good or are they bad? And if so, which ones are good and which ones are bad, and what can we do to keep them from showing their toxic effects? So they're toxic to tissues. They're controlled at several levels, including transcription, activation, and inhibition. And these are also naturally found in the body, and these are called tissue inhibitors. They're the main inhibitors of metalloproteinases. Now, aside from MS and the experimental model EAE, we've also looked at MMPs in stroke. So and also with just direct intracerebral injection. So MMP-2 was injected intracerebrally right into the brain, and it was shown that it opened the blood-brain barrier. TNF-alpha injected into the brain, remember that was the cytokine, into the brain, induces MMP-9 synthesis, and that caused delayed opening of the blood-brain barrier. So they kind of have dual roles. So in the stroke model, adult rats were given a middle cerebral artery occlusion for 2 hours. They were treated with a synthetic MMP inhibitor, which is called BB1011, and what they showed, they looked at the effects on cerebral edema and the blood-brain barrier, and this was done in Dr. Rosenberg's lab. Other ways that you can look, aside from looking at whether they're present in the spinal, in the tissue, and what effects inhibitors have, are looking at them in the spinal fluid. And so that's the goal, really, of looking at spinal fluid. It's an easy substance to look at. It's not easy for you to give. And so we want to use the spinal fluid that's already been collected at the center, if possible. But what we do is measure them with this process, which I already mentioned, called zymography. These, the tissue inhibitors can be measured by a process called reverse zymography. And more recently, something called ELISA, an enzyme-linked immunosorbent assay, has been developed, which is much easier. So we have some fairly easy ways to look. I don't need to go over DeVix. Everyone knows what DeVix is now. But Dr. Mandler, as I said, and one of our former residents, Wasim Ahmed, and a few other people in the lab, and Dr. Rosenberg, looked at these in MS and DeVix. And what they found, there were 7 patients with DeVix, and there were 23 patients with relapsing and remitting MS. They found that MMP-9 was elevated in MS, but it was not elevated in DeVix or in controls. So our question really now is, what about TM? Is TM more like DeVix, or is TM more like MS? And what are we going to find when we look at this? Because it may be an avenue for treatment if we find that there are differences. The tissue inhibitors, 1 and 2, were similar in all 3 groups, in the controls, the DeVix, and the MS patients. So the conclusion by Dr. Mandler, who can tell you himself, is that there were differences in MMP-9 supported different pathological mechanisms. The inhibitors that had been used included these with initials, KBR7785, which inhibited MMP-9 and MMP-2, the 2 gelatinases that we were talking about. Mice with intraluminal artery inclusion, so these back to the stroke model, had injection 30 minutes of inhibitor, this KB, 30 minutes before occlusion, and that decreased the MMP-9 activity, and it decreased the infarct size. So we don't know that all TM or any TM is really caused by ischemic changes, but we're suspicious that it is. And so we may be able to use this information from the stroke model in looking at inhibition of transverse myelitis. They injected at 1 and 4.5 hours, and these also decreased the infarct volume. So the results of the inhibitor were that there was reduced blood-brain barrier opening at 3 hours. Initial opening occurred at 3 hours, which correlated directly with these MMP levels, gelatinase A, and it reduced the brain edema at 24 hours. And so again, we've talked a lot about swelling in the spinal cord, particularly in some of the transverse myelitis cases and in DeVix, maybe less so as far as inflammation and edema with MS. No effect on either the blood-brain barrier opening at or the brain edema at 48 hours. So it seemed to be a transient effect, seemed to be fairly early. Other inhibitors, well, I already told you about corticosteroids. Aspirin has been used to inhibit MMP-2 in tumor formation. So we don't know whether there's any role for sure in the spinal cord. And I already mentioned the KBR7785, which you have to look up, and BB1011. So those are important potential inhibitors. What about in acute transverse myelitis? And that's what we're all really here for. There are unknown levels of MMP and TIMP, the tissue inhibitors, in the cerebrospinal fluid. And there are unknown effects of these inhibitors. What else do we know? Well, Duchessoy recently published this article in Neuroscience of scar tissue in injured spinal cord. Scar formation in spinal cord was accompanied by remodeling of the surrounding extracellular matrix. MMP-2 and 9 were found transiently upregulated in the spinal cord wound. So these apparently are showing up even in the tissue itself. And in situ fluorescent zymography, that way to assay for MMPs, revealed gelatinase activity in the wound that was spatially and temporally, timing-wise, correlated with scar formation. So these are there in lesions in the spinal cord. These were found along interwoven pathways along which the neurites, or the active nerve cell processes, were growing. These corresponded to distribution of other molecules with antagonistic or opposite effects on axonal regrowth. These neurite ingrowths may benefit from upregulation of the MMP. So can we, even after the fact, can we influence the activity within the cord with any of these either inhibitors or with the MMPs themselves? So our proposal is fairly basic. What we'd like to do is to look at spinal fluid in acute transverse myelitis acutely, because that's when it's obtained, and perhaps at a later point so that we could see whether there's still continuous activity going on. We'd like to compare these results to those already found by Dr. Mandler and his colleagues in MS and in DeVix, but also we've had several presentations here on infection, and we have cases that we are quite certain are associated with viral infections. So it would be very interesting to see if we can sort out those causes. And just in summary here, this is red chili. This comes from New Mexico. I thought I would use this as sort of a model to just summarize again. So if you have red chili and it is in the right place, like in the right foods, and you're asking to have this, it's pretty good stuff. But if it's in the wrong place, for example, it gets in your eye. Has anyone ever had red chili in their eye? you don't forget it. It burns. You feel like it's developing. You're developing a scar in your eye. And so the idea is that can we identify in transverse myelitis, either spinal fluid or in the tissue itself, whether these chemicals, these endopeptidases exist, and if there's any beneficial effect of inhibition of these chemicals. So I'd be happy to answer questions later. I think we have our next speaker. Thank you.